Okay, ladies and gentlemen, you've been hearing us talk at you for a while. We're going to switch back to the hands-on mode of the workshop. Under the direction of Vaibhav Pai. If you don't know Pai, Pai is a research scientist at the Allen Discovery Center at Tufts, where he's currently using synthetic biology and biobots to study a few things. First principles of morphology, basal, non-neural, somatic cell cognition, decision-making, and computation. Anything else? That's enough. Okay, that's enough. Awesome. All right, Pai, the floor is all yours.
So hope you guys are all excited about the biology lab. This is going to be interesting, particularly for me, because you guys all are going to be part of the discovery process. This has been never done before. So even Mike and I don't know what we are going to find out. So it's going to be interesting. So I've sort of divided into first half hour and second half hour. So in the first half hour, I'll have everybody come and walk through 3 stations that we have. So the first station is going to be xenobots for everyone to see. They are about 0.5 millimeter in size, and they have made the journey of 200 miles. So they are a little jet lagged. But you'll have to probably stare in the microscope for just a little bit. And if anybody is not familiar with microscope, just to let you know, you can move these 2 eyepieces to fit your eye size. The big black knob is for focus, and the short black knob is for magnification. You can zoom in if you want. So that's the xenobots. Then the second station here is going to be, as Mike said, some examples of agent shell material. So these are not xenobots, but we have here 2 kinds of organisms. They are both single-celled. One is stentor, which also moves with cilia. It looks like a trumpet. And one is paramecium, which kind of looks like a fruit slipper, but they are super small in size. The stentors are not happy that they are super away from home, but the paramecium are just loving the new environment. So you're going to have to dig a little bit deeper to see the stentors. They are a little bit reddish in color, but you can clearly see them by their trumpet shape, whereas parameciums are going to be these small white thingies that are going to be moving around. And the third thing is here, which are the planaria worms that you saw in Mike's talk. So they are always camera shy, so they are always going to be around the edge of the dish, but I think you should be able to come see that. So in the next half, what we are going to do is we are going to have a planaria Turing test that we will do, and an interaction between the agent shell material and xenobots. So more on that I will talk in between during the 2 sessions. But why don't you all one by one come and go through these. I would say that those who have already seen the xenobots, skip this station so that those who haven't seen them before can get a chance to look at them. And I would say stare at them for about 15 seconds or so to see their motion, because they are a little slow today.
I can help run. I can be that would be great.
It's like a centaur, maybe.
And if anybody has the heebie-jeebies of dealing with biological material, I have some gloves here, so you can feel free to use them. There is small, medium, large. So
I've already been injected with xenobots, so I'm fine.
Yeah, you can use this for magnification. Each person's eyes are different. So if you want like really zoomed in, I can
really zoom in. Okay. No, it's still not. I want to see the stumps. Yeah, the molars.
Your vision's going to tell me.
You're the go-getting xenobot, I'm the waking up the zombies.
Some of the vibrations that you're going to see.
That's in the inner room.
Okay. I think this is as much as I can zoom in on this scope.
$10 million.
Fine, we have a really active one area right now.
Yeah.
There's one, they're all active, one that's sort of floating around, and one is they're not super, real happy.
I mean, they generally like to be around the edge because they don't like light as much. So they try to stay away, so they try to stay on the
Shaky, okay.
Yeah, be careful because the cables aren't stable, so if you put any weight on them,
Peanut butter everywhere.
no, it'll just make most people dizzy.
Wait, where is IPZ?
So this is up and down for focus and magnification, and if your eyes are thinner or widely hyper.
Oh, I see, just like that, okay.
Sometimes it's easier without glasses. You do as
Is it super zoomed in? I can zoom out if you want to see a lot more of that. Yeah? Okay. Just they're quite slow today, so you have to hold on for 15 seconds or so, and then you.
I think transporting them actually made them like.
I think they are, yeah, because also it's been warm
Yeah.
comparatively, so what that does is that speeds up their aging process.
Sure.
So,
They're starting to senesce.
Yeah. No, these are actual.
If you jump into a pond, you will be
What, so deep?
Yes.
But what you will make is see how they interact. Yes, you'll see that in the second half.
Hi, can you help me for a second? I'm just not getting them focused.
Okay, so focus is this thing actually. This one is for zooming in. What I can do is I can refocus them for my eyes, and then maybe that will give you a ballpark area. Okay, great. Oh, it was just during transportation I was trying to protect. So I use all that stuff.
You're doing it right. By the way, I met their whole out of that vapor.
Right now they are at the bottom of the dish. You should see them as kind of a little bit reddish. They have like cilia and they have cilia over here. Sometimes you also find them moving in the water. Not necessarily. Sometimes they are. Sometimes they can be just moving through the water.
And you're running there's many ways you can go
Slow motion,
That's easy, right?
Everyone just leave.
Yeah, he's barely flopping around. This is a good owl, though.
And then if you move them up to the water column,
Office investing in stuff.
I would say do this, obviously you can see a lot of them. So the trumpet shaped ones, you see all the way at the bottom, and the area ones, you can see floating in the water.
Just raised a bunch of money and we're still trying to figure out what they want to do.
So they only have it for the summer. And then where does he go?
It was an unsatisfying answer.
What's that?
It was an unsatisfying answer.
To one question. I mean, all of them were, but to which question in particular?
When the patterns are established in photosynthesis.
I mean, you want the real answer? Yeah, it's before the universe got going.
Yeah, okay.
You have a little jet lag.
When did your patterns trying to break through into this dimension idea begin to emerge?
I think everyone
Having the
Is recent or not so recent?
1983, 85, but I couldn't say those things before.
Feels new to me.
Yeah, well, in certain ways it's new, in certain ways it's very old, but long before me, but I couldn't say those things until recently.
What is
stare at them for about 15 or 10
difference
seconds.
between
I think for your eyes, you might have to adjust this.
if you posit that the creature has a goal, tissue or unit, atomic unit, and will seek to achieve that goal by whatever means.
what's the dollar associated with phase two?
After everybody's done watching,
What's the coding for the, let's pretend the coding for the goal. Some pattern.
Sometimes it's by electrical, we see it, sometimes we see it.
Well, what is coding for the, not the methodology? In other words, how does it learn? Does it create methods, or does it, is there, or is it just saying, I have this goal, and I'm going to do a bunch of dumb behavior, novelty-seeking behavior, but what's the, I know I have a goal, so.
Sometimes it's second order frequencies.
Do you think that phase two should ultimately be continued or disruptive to find a different solution? Do you think that might be worth it?
I think it depends on the outcome.
And sometimes
Because I really worry about
so for example, like I might
forcing it to go down that road. But I worry about the core technology ultimately.
So
there may be expertise, but there is expertise needed beyond what they can do. That gets related to the project. On the vision, they don't have the expertise to have people.
Yeah, I think stare at them for about 15 or 20 seconds because they are a little slow right now. So, yeah, but you can see them. Are you able to see them?
The behavior.
Those first ones were just.
Yes. So Xeno was obvious. This is the agent material. We have two of them here. These are single cell organisms. We see them move. And in the second half, we will be doing an experiment where we will be seeing interaction between them and xenophiles. So for now, we are just seeing them as they are. And they're on the third station of planaria. So in the second half, you'll be seeing interaction between planaria and xenophiles.
This is sort of the original product. This is our testing.
Yes.
We're going to learn that you can indeed monkey with the OEM.
Yeah, and then can we find out a way to convince them to be integrated with xenophiles? To create some kind of additional motion, right? Because they already have cilia and they already move, but they don't move very differently than xenophiles. They have a different metabolism, so their movement patterns may be kinetically different, as well as shape and direction-wise different. So can we then combine those much smaller, much smaller? These are single cells. Yeah. The centers will be
Very dramatic. It's happening now.
This is all happening as
So do these guys, they chase food or do they
Which ones? These will chase food. These quite often times will have their hold and will stick to the bottom, and the cilia is on their mouth, so they will draw food in. But a lot of the times you will find them just floating around trying to catch food as well. So they kind of go both ways.
Even the idea of healing, they say that they use
Yeah.
I see you just grab something a wave.
Sometimes they do. I think they definitely good. They have the ability and to move this to go up and down the water column.
My mic is always wearing the face mask. I don't know.
Because they really put nerves in it. They can stretch it because it's kind of a good time.
Did you publish them about the news?
Using this, you see what this you will find at the bottom is Anything dead and decaying matter or even smaller rivers that are there like Daphnia and other things. But sometimes even like big giants, sometimes they will gobble it. Yeah, glasses will make it worse. Try to look at them directly and stare at them. Yeah, this is the bottom, all the way to the bottom, you see the spectacles at the bottom, and then move up again to the water bottom, and you see the parameciums in the sometimes they don't do anything, it's just one direction. But majority of them will go in circles, and then there are some that will go in circles, some that are partners. So generally they go one of these 3 or 4 depending on the shape. So land exactly the shape is very hard to do. They any slight changes based on the orientation of the so that will give the less randomness in the next slide. So what? Since everybody can't, so I'm going to do the experiment in the next half. Once everybody's done watching. Since everybody can't watch through the microscope at the same time, what we're going to do is hook these up over here and live stream them on the screen, so you can actually watch the experiment happening. So we are trying the AVs thing. Hopefully it works out.
Well, it almost certainly will not.
And the bottom you will see these to the water color.
Oh, it's amazing.
Once move all the way to the bottom of the dish, and then you can see these rocket shapes right They have some amazing and on the bottom side. We kind of go to things like this.
We have a lot of there's a lot of demand for
So I had strong opinions that the way it's very but one of the reasons that for ways to build AI. This is great. I'm glad you guys are doing it. Really internalize
Sorry.
So many things. So the way that I would start meaning that around 4 average and eligible to what Mike has found is
Yeah, when you market something.
so you don't have to mix around DNA, you don't have to change the just incentivize the cell.
That's the food. The green stuff, right? Yeah.
Some not constraints.
Oh, by the way, did you ever see what
I brought them on Sundays and from Boston. So that's why I was not sure they were able to survive. That's why we are a lot less in number here than
Some ways, really, I think that's extreme.
Very much the same way that cells tissue. So you can actually show in some different
Useful for the sales.
really good.
Literally.
Cells.
Free for sales and
Switch
on the camera.
So right now I will show that movie and then want these on, but I'm going to start the experiment anyways right now.
Yeah, I take them like wire. When they walk, I think I know.
They are, yeah. So it is. It's this the thesis I have is which entirely relies on human data for images, for image generation, for video. Which does not work out so well. Yeah, and so that paradigm is very classic. Very skeptical. And so there are so many reasons.
That's very. Oh wait, that's a good one.
Working on that.
So he and Doug and Patrick.
And so there are
they use an eyebrow.
But the reason I will stick to this, there are many, but one example is
Literally pluck your eyebrow and make a knife out of it.
So the main difference
Just strong enough, but just if it's too hard, it's too soft.
and achieve superhuman results rapidly. AlphaGo. AlphaGo was top. Right?
So you go to the level lab if you want to work on the bots, you should test your eyebrows. Of course.
AlphaGo 0 was nothing.
You need to contribute.
Yeah, if you get that eyebrows, you got to cut them off and get them to the lab.
Not knowable, not provable. The only data we have is empirically, whereas AlphaGo 0 shot past that. So that's one small data point. We have many others. Generally, the idea is that human data limits to the human paradigm. There are other paradigms out there. And so, if we want to actually discover those, we have to be able to do this in an efficient way that's not relying on human data. Good version. Yeah. good version is kind of like what
Yeah.
talk about the property.
Yeah, the paramecium is like centaurs and they're selecting as many centaurs as they can because they are generally the ones that
creatures that have their own hierarchy.
my guess.
More than they will not speak English as a first language.
I think this one is ready to stream. So for streaming, we will use the same screen, like shut that off.
Or
you could have different screens, like for streaming, we
something like a
switch on all the
proxy
TVs.
system.
Actually, it's not necessary.
It's learning English as a second language. Make sure that don't learn right? Give it these moral language.
Thank you for your emergency assistance.
Oh, yeah.
Professional emergency assistance.
Not terribly. A couple steps from this. So, Stephen's talk is adjacent to one topic I'm very interested in, which is how do you mathematically capture all these ideas like how do you capture these ideas of molecules forming into a cell, cells doing that at the next level. Take those mathematical notations and
Because I gave you my number. And said, send me a text and I have your number, but I never, so I was just looking for yeah, it's a little bit late, but then I, ordered it. I looked at what I could do, because I actually have really too much. And then I also actually it was even more ways to that, because then I can more things I could slowly in my own.
exactly. Yes. Yeah, so I have one program now that's in this area. I'm working on a much more ambitious one, which they may or may not fund because I'm asking for all but
Probably not.
Like it's expensive, and then they can like.
Okay.
Yeah, I've been collaborating with Tristan and just realizing about. This is exactly your point is, which is that there's a huge higher overcome and apply to other things what you get out of a large scale flat latent space of a neural network is not that, and you can have tiny perturbations that input that aren't meaningful to the human that resolve and radically which makes them not suitable for a level of so they get answers right sometimes, but not in a systematic way that reflects some underlying knowledge structure that we really want. So I think we talked about, which is kind of breaking the I think we have composing would be more reflective of how we learn. I'm not necessarily like going, I think even if we just level intelligence, that would be incredible, and I don't go anywhere near that. Like, I think we're a lot more amazing than the questions and things that we think are like global direction.
Yeah, but it's also interesting.
Okay. I'm gonna go.
sure. Yeah.
He's on.
Yeah, okay. I told that you did this structure. I told that you would. Yeah.
Not start recording yet. You show the video.
No, I want to have my chance. Did you find everybody in there? Yeah. Okay.
Oh, you're getting a sneak peek, Josh.
Oh yeah.
Because I'm about to say, yeah.
Okay.
Now you're going to see a mixture of xenobots and the other
Oh, I
organisms.
see.
So we're going to live stream this.
So I
So, and you'll see,
So am I looking at the Turing test or the fight club?
you're looking at the fight club. The Turing test is going to happen here.
He's trying to address the question of how fundamental is
Oh, yeah, it's totally
and
different.
one of the experiments.
So what I was hoping
you see it in the Stentorius?
Yeah, they hear that? Showed that the story does a lot that we never even thought. So we only pay attention.
Yeah.
Mike, I
heard the word bubble sort. Hairs on the back of my neck.
How did you want to take guys over?
And you're a pro now.
Anything wrong. So it finds that bubble sort, of course, it sorts the list that shows all of these computational side effects at the same time and doing, fairly high complexity interesting things that we never noticed because we were never looking for. And so the more general question that I think Mike is interested in is where does compute happen?
So you go in once a week?
Yeah, okay. Than just what we there.
All right, guys.
Yeah,
stop looking.
Yeah, there are a number of others since bubble sort of developed a number of other
Otherwise, we'll be here eating up Hale's time.
simply not in the algorithm.
Yeah.
So, right, so what do you mean by like time?
Alternative.
So it's I'll send you the link. It's kind of a long story.
Cut into your time. We'll just cut into lunch.
That's two steps of the algorithm. There are specific things that the algorithm is telling you to do. So all the numbers you can look at it from
And maybe yes.
It's facing the other way. It's optimizing other kinds of metrics that are not represented in the algorithm at all. So,
Are we going to be in that lunch room for the rest of the afternoon?
Yes, we're going to stay in there. The acoustics are not great, but
you
we'll.
won't know that. We all know that.
Worst case, I can just carry my laptop around and other people.
Yeah, that's right.
As I walk.
That's fine.
Yeah, so we just did it by taking a
Also, Caleb's
behavioral science approach.
Oh, great, okay,
He's
yes.
not physically there yet.
Okay.
And again,
In the meantime, I will.
Oh, awesome, okay. I'm not biased at all, but I think you made the right choice.
No, I am very, very.
Hey, everyone.
Good.
Can I have your attention here? So we have started an experiment, a couple of experiments over there. One of the experiments is where we put planaria.
Attention to the front, please, attention to the front.
Hey, everyone.
Eugene Durenard, aging Eugene.
So we have started a couple of experiments which we will be streaming live here. What I'm about to show you is not live. This is a pre-recorded thing, but just to prime you guys. So the two experiments that we have started is we have put planaria and xenobots together, and the paramecium and stentor and xenobots together. But I wanted to prime you with this video where we have put planaria and xenopus embryos together just to show you what they do. And then I'm going to take bets on what you think is going to happen. This is, by the way, a 2-hour long movie that I've sort of sped up for 2 minutes. So it's only going to be over 2 minutes. They obviously don't move that fast. So what do you think is happening?
They're eating them?
The region
Yeah. They're eating the xenopus embryos. And within 2 hours, you will see that they clean out the plate.
Well, at least they're polite and finish their dinner.
Exactly. Now, the real question is, what do you think they are going to do to xenobots? This is the Turing test, right? Are they going to recognize it as a xenopus and eat them? Or are they going to recognize it as an entity that is not xenopus and maybe be curious but not eat them, right? What are the bets? What do people think?
What's the size difference?
I would say maybe half to a quarter. Xenobots are smaller than they are. Yeah.
But they're still hungry.
Yes, they are hungry right now. Yeah, so this is not a video that I captured right now. This is like a video that I captured just before coming here last week. So they are definitely hungry.
And Pi, are they these the Xenobus here? Is it the same, more or less the same age as the Xenobots that were?
No, they are younger actually.
These are younger than
Yeah.
the Xenobots
Yeah.
we're about to see.
So the siblings of these embryos are the ones from which I made the Xenobots.
I assume you already know the answer.
No, I don't.
Oh,
That's the thing. Like that's why. Like I don't know the answer of whatever we're going to find here. So okay, this movie is just about to end. So as you can see, they clean out the plate. So now we are actually, Krishna, can you switch to the?
Can you fix the cam?
Yeah, this cam is already on. So this cam you should be able to get. There is some.
What are they? Oh, I'm sorry.
Yeah.
What are they not? Any priors?
I think they have. They definitely are picky. They love what, beef liver, right?
They love liver, they like
Yeah.
egg yolk,
Yeah.
there's a few things they like. They sometimes attack each other depending on the species.
Yeah. But they are picky. They don't eat everything.
No.
Yeah.
So there are plenty of things in the size range of xenobots
Oh,
that
yeah.
they would not.
Yeah.
Do they eat plants?
No.
I'm just curious now.
They
Has nothing to do with this.
They don't.
In the feed. I don't know where it's coming. But I'm not sure. Oh, there it is coming. Okay.
There we go.
There you go.
All right.
Oh, if you turn the light off, you're not going to get anything. So don't turn it off. Yeah. I'm going to do like a little MacGyver thing where I'm going to put some things on top of here so that we can get a better. Removes the light reflection.
It's in the box, yeah.
All right, let's see what happens.
So what's on the left, what's on the right?
So these are planaria and xenobots. So these are xenobots. These are the planaria.
Yeah.
And then here you have the xenobots. The white little things are the paramecium. And then stentors are somewhere floating around. I know this resolution is not great, but that's the best we can do for now with live streaming. And yes, this is going to be much slower than the movie. The movie was like a super sped up one. So we'll see what happens. So we'll wait for maybe another 20 minutes or so. Oh, what?
It's going for it. Well,
Did he eat it? Oh, he's smelling it. He's smelling it.
yeah. See?
Nope. Didn't take it.
Whoa.
Amazing.
So they like the embryo but not the xen.
We don't know. We'll see.
Do you have one of the xenobot embryos that we can use for control to make sure they're actually hungry?
Oh, I couldn't bring embryos because of, like ICOC and other reasons.
Oh, fair
So, yeah.
So explain, so the first video that you showed was what? Was
Xenopus embryos and planaria.
that the planaria eating the
Embryos.
the embryos, okay. Not the xenobot.
No, not
Yeah.
xenopus.
I see.
We had never tested. Even that, I just did like a day or so before coming here. So yeah, we don't know what's going to happen here. They're definitely interested. They're going and smelling.
Yeah. They keep going up to them.
Yeah.
Why do they have noses?
Yes, they have some olfactory stuff all along the top edge of the head. So if this is a planaria with here the eyes, if they have an object that they want to interact with, they will go up and like kind of smell around.
Where did the other planaria go?
Still on the edge.
Josh, you might get a better picture on here, sharper picture on here.
You mentioned the eye stuff, as a concern for the ethics of the embryos.
What do they make of the xenobots? Like, do you have to have
No,
meetings with them or they? Oh, okay.
So far.
Did that one just eat one or is it just pushing it around?
It could be both. They are playing, maybe.
They're definitely pushing him around. Is he eating it?
If it goes over it and it doesn't show up at the
Yeah.
bottom, then that means it ate it.
I think it just ate one.
Oh, did it?
I'm not sure.
No, they've gone up to a bunch of them that actually exist. Don't want them at all.
One disappeared under the.
Nope,
oh no,
nope,
no.
No, yeah, they're.
It just pushed it around.
Yeah.
But I mean, do you think that peer
There's one that went up to several.
Yeah.
As a matter of fact, turned away immediately.
Very good. You want a minute for me? Sure.
But why are these robots not moving? It's because they have their.
Well, they move actually. So the videos that Mike showed, they were sped up. At least, so in those videos, every second was actually a 1 minute. So they move relatively slowly, and especially these are a little traumatized from the travel and and they were a little bit in the hotter environment. So what happens is if you keep them at higher temperatures, they will age faster. So that is also there.
Zero interest.
Yeah.
I mean, there's interest in them, but not in them.
Yeah, yeah.
Given that they're made of the same stuff.
Same stuff, same cells. Nothing is different, right?
When you make the xenobots, isn't it you're taking a slice off the top of the?
Yeah, I mean, it's not all of the embryonic cells. We are taking maybe like 20% of the cells. But still, they are.
But those cells, they're not the yolk, right? Because Mike said that they like the yolk.
Yeah,
so that's probably-
yeah, could be. Yeah, absolutely. Interesting experiment nonetheless.
I think they.
And they have their stomachs on the bottom side, so that's why you may not be able to see them eat, but if it disappears from the bottom, then you know that it went in.
They just don't like that.
Oh, it didn't eat it. Still there. Instant replay, let's see.
Krishna. Oh, Krishna. Are we recording this?
So do you have any idea what the difference is? Like if they're made of the same stuff, like why would the...
My guess, if they are not recognized, like if they are not eating them, my guess would be the yolk.
The yolk?
Yeah. So they are looking for basically fat.
I would have bet money that they would have just chomped through them.
That's what.
I thought this was like, I don't know, kind of a constructive little demo because I was like, oh,
No,
they're going to eat them, but no,
I'm
Curious how if that will happen here, like, on the other hand,
How long will they survive at this point?
Sorry?
How long will they survive at this point? Like if you don't.
The xenobots? The xenobots or the planaria?
The planaria.
The planaria will survive for a very long period of time. They can
Even
survive.
unfed?
Unfed, they will survive for several weeks. What happens is that they eat their own cells, so they become smaller and smaller and smaller and smaller and smaller, and finally gone.
I wonder if eventually they'll.
They die eventually.
No, like in this case, I wonder if you left them, would they die out or would they eat first?
Yeah. So they are already starved for 3 weeks now. So this is like a really hungry planaria. It's not generally for the.
But you've just exposed them to them, right?
Yeah. Yes. So generally, what happens is so in the video that I showed you about the Xenobus embryos, same. It was 3 weeks. I put them in there immediately. They went outward, right?
Yeah.
So.
But that's a known food source for them.
Not necessarily. Well, maybe. Yeah.
I mean, they know they like.
Yeah.
yolks, so I think like if I'm sitting there hungry, like, I gotta get really, really extra hungry before I'm gonna eat something I don't like. But eventually, I'm going to give in. They're just really tasty.
Guess, because we don't know the real answer, my guess is that they are going for the yolk in embryos, which is fat, which they like. So they like beef liver, they like, those kinds of my guess is that most won't have yolks. they're smelling them and they're like, oh, this looks like, it's kind of like, think about it, you're getting fish, but in this case, they are getting only fish skin. So they are like, they're going and smelling it because they can smell it, but then they're like, oh, there is no meat here.
What happens to the material that they do ingest, like when they eat egg yolk, what happens to it? They just turn it into energy?
They turn into energy and their excrement, their ingestion and excrement is through the same organ called pharynx. So they will take it in, process, digest, they have a digestive system and then they will excrete out through the same pharynx. Owl? Really?
They digest things and turn it into a pellet and then they spit it back out.
Really? No, I don't know about that.
But do they, so they have one?
They have one opening for both entry and exit. And it's on the bottom of the it's on the bottom side. And when they get super stressed, they will actually release their pharynx. So I'm almost like it. So here's another part about the agential material. The thing is, the pharynx that they release when they are stressed still moves around and eats stuff, even though it's not connected to the planaria. Which is bizarre.
But if you cut them after they lost their pharynx, do the new ones have a pharynx?
Yeah, they grow.
Oh,
And then, like, it has like 6 different types of heads?
Yeah,
So like,
those are 6 different species.
species, okay. So it grows the same exact shape after?
Generally, yes. If it is unfurthed, it will grow the head of the exact same species. But a postdoc and a grad student in the lab who worked with planaria, what they were able to do was use bioelectrical signals without changing the genome. They were able to actually have the planaria make the head of another species that was about 6 million years difference in evolution. Which was really remarkable.
There's a owl product, so you can tell what they've been eating.
Oh wow, that is crazy! I had no idea about that. Amazing.
Yeah, I think they
It goes in and out the same.
But that's like the hair and the bones, right? And they also.
What are you talking about?
Just because the worms.
And then, like, I can understand why some what's about that information in it, but not expressed, then is triggered to then create.
Different people will have different theories, so they generally have a set of cells which are called neoplastic and quote-unquote stem cell-like. So there are certain people who think that, oh, it has to be the neoblasts, and what they have done is, this is not our work, this is other people in the planaria regeneration field, where they have essentially irradiated to remove kill all the neoblasts which get killed by radiation, and then transplant a single neoblast from another worm into this irradiated worm, and it still repopulates and reproduces. And that's one theory. What we think is more nuanced than that, in the sense that it is bioelectrical signals. The reason why we think that is because even without changing the neoblast or anything, we can change the complete morphology of it just by playing with some ion channels, where there are these distinct signals for head and tail, and if we can recreate the signal of head instead of a tail, it will make a head there. So again, we are not touching the genome, not doing any genetic engineering or any cell transplantation or anything like that.
And do you have instruments understand which genes in the biochemical signals and then before what biochemical signals they're not?
planaria in that sense are very hard to work with because a lot of the stuff is unknown. Like you can't do most of the molecular biology, like you can't do immunology in planaria because there are no antibodies that are there. So in terms of fine detail molecular biology and especially biochemistry is harder to do, but I think this field is slowly catching up.
Something that might
Stop. Yeah, that's the biggest question, right? Right. So there is an internal representation of some sort that it is constantly checking. Am I correct? Am I in the correct morphology? As long as it is not, then it will keep doing the error minimization schema. As soon as it gets okay, now it's exactly the same as my internal representation. It stops. What that is, we don't know.
And there's no way yet, and maybe there is a molecular biology that representation.
That's the biggest part of our work right now. So we don't know yet what that is. I mean, we think that it is bioelectrical signals, at least to some extent, contributing to that internal representation. But there could be biomechanical signals. There could be others as well.
You know, this really reminds me that reflexive and so like thing that's happening inside the cell, like when you put the organism. That's like, okay, I'm reflexive, like I'm not going, I don't need to, unless it's like an anomaly or whatever, I don't need to grow my nose and work with my knees to do that. So when you cut off, and the person draws on that's instead of just this.
No, that's a very good hypothesis. We will probably need to test that. But
And I think with the non-women, like they are. This is just so different. That's just different with the non-women self-replicating.
Right.
Very different replication. Because it's there's no conversion. It's only reflexive.
And at that point, if we don't provide it the raw material, it's going to stop replicating. Right. So.
Thank you. Yeah, awesome. Yeah, thanks. Amazing. Yeah, I'm interested in like kinetic replication.
Yeah, that was like von Neumann is outside my sphere of expertise in computational robotics and all that stuff. You could have machines theoretically that replicate themselves and then make more machines and so on and so forth. So this xenobot replication is that, but it's not reproduction that is classically, because there is nothing inherited. There is each one is different completely. It's not clonal at all. And it is based upon your input. Because if you don't provide it the raw material that is the cells, it can't make it. So there are pluses and minuses on both sides.
What other raw materials do you begin?
That's the only one we have tried so far, but this is another example right now, right? We are putting it in along with paramecium and stuff and seeing what happens. So,
how does it work? So you add cells as raw material, basically just complicating.
So what it does is some of them, so they have several different movement behaviors, and some of them what they do is they move in circles, and as a outcome of that movement in circles, they basically generate piles of cells, and once those piles of cells happen, they automatically clump together and form a xenobot. So and then that xenobot will again start moving and make another pile, and so on and so forth. So I think so far we've seen 4 generations of those. So that's basically how they quote unquote reproduce or replicate.
And then we're going to see agental materials. This is what like.
So we are calling these paramecium because they are essentially a organism by itself, right? They have their own agency. They do their own thing. So that and the stentor are basically what we're calling retention material, because what we theoretically in the future could do is integrate the paramecium and stentor into the xenobots. The reason for doing that is because they have very different movement behaviors, very different metabolism, very different kinetics. And so if we want to achieve, say, X, then we can do these combinations to achieve that X, right? In terms of whether it's motion behavior, whether it's changes in the metabolic space, whether it is change in whatever. So this is just the beginning of that.
So maybe you're saying that the xenobots would take in?
We don't know what they will do. We have no idea. This is the first time we've done this. So we are also recording this. So I'm going to watch these for a very long period of time, you know.
Yeah, and get the AI to watch it.
Yeah.
Computer vision.
Yes.
Where's the them from? When you need them.
Yeah, I know, right? But yeah, I'm sure somebody in Joshua will do that.
That little tan one's been pushing that one around a lot.
Yeah,
Oh,
it's
yeah?
small. It's so hungry.
Yeah, he keeps circling the same one.
He really wants it.
Fine, I'll eat it.
How many xenobots were in there?
I think somewhere around 10.
Okay, I've counted 10 this entire time, so I have not lost any of the ones I've counted.
Okay. I think what I'm going to do is, because it's already running late, I might just, can we keep the recording going and then have Kali doing her presentation, and then we can just look at the extended video later.
What's recording then?
That computer over there?
I might need that for Rick's presentation.
That's what I was asking him.
Oh, there's another computer. Okay, great. Yeah, let's do that.
Because we are already running late, right? So
I'll project it on these TVs, I guess.
Oh yeah, that's great. Hi everyone, can I have your attention? So the experiment is going to keep going. What we are going to do is, because it's already an hour, it's what, 11:37 right now. So we are going to keep the experiment and we are recording it right now, and it will be displayed on those scenes over there, but we will move on to the next presentations and stuff so that everybody is on time with respect to lunch and all that stuff. And then maybe later on, I can show you guys the recorded video just sped up so that we can see what the interactions happen.
But what do you expect?
There is no expect. We don't know what to expect. We don't know. We have no expectation.
Let's take a quick poll. Take a quick